Our next new fellow is Professor Mark Smyth, who's a highly cited immunologist who is recognized for his significant contributions to tumor immunology. His research has helped pave the way for effective immunotherapy of cancer, beginning with immune checkpoint inhibitor drugs. Mark has also made substantial contributions to our understanding of natural killer cells and T-cell biology. Thank you, Mark.
Thank you very much, TJ. Thank you for the opportunity to speak to you today and for the honour of membership of the Academy. I'd particularly like to thank those people who facilitated my nomination. My career has been focused on studying whether there is a natural immune reaction to cancer and whether that affects tumour initiation, growth and metastasis. And this passion of mine has really been fuelled by Macfarlane Burnet and Lewis Thomas's original cancer immune surveillance hypothesis that they formulated in the late 1950s. And the idea really was that lymphocytes had the ability to recognise early transformed cells and could surveil, recognise and eliminate those. And this idea really went through a roller coaster of ups and downs in terms of approval or disapproval based on experimental evidence for over about a 30 or 40 year period. Then we entered a phase where gene knockout mice became available and sophisticated tools for knocking out or neutralising proteins, particularly in experimental mice, and I used those systems to really exploit and test that hypothesis. And I'm glad to say now in 2017 that immunotherapy is considered the fourth pillar of cancer treatment to surgery, radiotherapy and chemo and targeted therapies combined. So cancer, as we know, is a massive global health problem. It's the #1 killer in Australia, and worldwide almost 8 million people die of cancer each year, about 1 in 2 men and 1 in 3 women diagnosed in a lifetime, and cancer deaths are predicted to rise by about 35 to 40% by 2030. And in 2013, Science announced cancer immunotherapy as a breakthrough of the year. Now, this breakthrough was really realised on some exceptional clinical data that's been generated in the last 5 years. But truthfully, it's been founded on at least 5 major areas of foundation work over about a 2-decade time frame. And these include the following. So cancer immunoediting, firstly, that's a revision of Burnet's immune surveillance hypothesis, and it was proposed rather brilliantly by Professor Robert Schreiber at Washington University School of Medicine, St. Louis. And that idea was underpinned, I guess, by data generated from my laboratory and Bob's laboratory. The second is the concept of tumour neoantigens. So this is a very old discovery. Tumour antigens were first described around the time of transplantation immunogenetics back in the 1950s and 60s, but weren't really isolated for some time. And more recently, this idea has been reinvigorated again by Robert Schreiber's use of next-generation sequencing and algorithms to demonstrate the expression and editing of tumour antigens in highly mutated tumours. The third area is human immune reaction, and this is based on old pathology, obviously, but revolutionised by Immunoscore and detailed human tumour landscape geography studies done by immunohistochemistry by Friedman and Gallon, the first in colorectal cancer, but this has been rolled out into many other cancer types. The next concept is combination antibody-based therapy. This is an old concept in cancer and HIV therapy, but it's been pioneered really by my group in immunotherapy at first and has been embraced by the pharma industry since. And the last concept, not the least important, is tumour-induced immune suppression. So this is the work of James Allison on immune checkpoint molecule called CTLA-4 and other groups on other molecules, immune checkpoints called PD-1 or PD-1, PD-L1, the ligand. And that really defined a layer of immune reaction, or regulation rather, that can be stripped back in tumours and has led to some very dramatic clinical results. So firstly, the concept of cancer immunoediting that Bob proposed. It's a simple concept that involves what we call the 3 E's: elimination, equilibrium and escape. Now, the elimination phase is really the original cancer immune surveillance concept, and my group and others have done a lot of experiments using knockout mice and neutralising antibodies to define various immune factors that can prevent the development of tumours or halt the progression of tumours. And so tumours can be killed or eliminated all the time on an ongoing basis. And of course, there's a lot of genetic changes that lead to transformation, and we're starting to learn more about how the immune system can detect these changes very early on. And this involves all different types of cells, innate cells and adaptive cells. Then there's another phase called equilibrium, which was really just a hypothesis, but has been tested, as I'll show you, where there's persistence of the tumour in the face of the immune system. And the immune system actually sculpts the tumour. And the tumour that eventually escapes has then become immuno-evaded effectively. And this battle can go on for a considerable period of time due to the genomic instability of the cancer and the ability of the immune selection process and many arms of the immune system to eliminate cells. So one of the experiments we did to test the equilibrium phase was using a carcinogen called methylcalanthine, which induces fibrosarcomas in the animals. We inject them with a low dose of carcinogen, and then what happens over a period of time is there's a group of animals that get primary tumours that we see grow out very quickly, and we exclude them from the experiment. And then we look at the animals that have got just basically small lumps on the surface that contain the corn oil in which the carcinogen's injected. And we divide those animals into two groups, a group that we treat with a control antibody or a group that we immune deplete using T-cell depleting antibodies or neutralising for interferon gamma. And the really remarkable result was that in the control situation, you can see that those lesions underneath in the subcutaneous space eventually disappear in the control setting. But in fact, there are tumour cells dormant in that place. And if you immune deplete out at, say, day 200, as shown here in the lower panel, you can see that suddenly there's a tremendous outgrowth of tumours at that place. And we demonstrated that in this model, and we demonstrated in other models like P53 loss mutation, for example. And indeed, you can leave animals for almost 2 years with these dormant cells in place and still immune deplete, and these tumours grow out. So this was good evidence for this equilibrium phase, and others have since gone on to show this same sort of biology in UV-induced skin tumours as well. So in terms of immune checkpoints, there are a tremendous number of surface interactions that regulate immune cells, and particularly T cells. And these brakes on the T cells are there to prevent massive autoimmunity and self-tissue damage. And so a number of these have been characterised, and I'm really just going to talk about two of these that have made substantial impact in the field of cancer immunotherapy, and these are CTLA-4 and PD-1, and they have slightly different mechanisms of action as we understand today. CTLA-4 seems to work more in the periphery. It also works in the tumour microenvironment, and it acts at the level of the priming phase to increase T-cell proliferation. So normally, CTLA-4 is a checkpoint molecule, if you like, that binds a ligand on the tumour cell, and this essentially shuts down the T-cell receptor signal, which is the primary signal that the T-cell needs to get. So by using an antibody to block this interaction, you can essentially reinvigorate the T cells and the priming of these T cells in the lymph nodes. The second pathway is a very similar type of process, again, where the expression of a ligand, PD-L1, which is regulated by interferon gamma, can be upregulated on the tumour, and the PD-1 is engaged, and this essentially shuts down again the TCR signal. And this particular interaction is pretty specific for the tumour microenvironment, which is really good. So in terms of generating antibodies to this interaction, again, this has very dramatic effects, and these are occurring in the inflammatory site. So to just show you the sort of clinical development that's been remarkable, and I guess very satisfying from my point of view, is in 2010 we saw the approval of ipilimumab, which is anti-CTLA-4 for the treatment of advanced malignant melanoma, an overall response rate of about 10%. That doesn't sound a lot, but for melanoma patients with advanced disease, this was a big breakthrough. We saw improvements with anti-PD-1, anti-PD-L1 reagents, so roughly 30% overall response rates and a better safety profile for that compound. And then we started to see combinations as early as 2013 with anti-PD-1 and anti-CTLA-4 together, and we saw an increase in the overall response rate in patients. And some of the more recent data, particularly in untreated melanoma, so as we're getting to treat earlier and earlier patients, we're seeing better outcomes with immunotherapy, overall response rates of 61% reported in 2015. Anti-PD-1 has demonstrated broad anti-tumour activity in a lot of different cancers. These are just waterfall plots to show you the downward trend is the reduction in tumour size. So you can see across a wide spectrum of cancers we're seeing clinical effects of these compounds. And you can see the massive expansion in clinical trial activity now. In the outer hemisphere, if you like, we've got the sort of early phase trials right down to the phase 3 in the middle. And you can see a broad spectrum of cancers where lots of different things are happening. So the change in paradigm is to really go from some minimal benefit with chemotherapies to actually raising this plateau of survival with monotherapy, immunotherapy, monotherapy, and even better effects with combinations. And the challenge is to raise this curve obviously further and further. With anti-CTLA-4, we have data out as far as 10 to 15 years now. Anti-PD-1, it's still early days. We also see a change in the way that we think about tumours now. So we've traditionally thought about them genetically, but we have also the concept of microenvironment. So the type 1 microenvironment in the upper left corner is the patients that respond very well. We call it adaptive immune resistance. The group we've really got to work on now are the immunologically ignorant tumours, the type 2s there, where there's no T cells there, there's no evidence of a PD-L1 pathway engaged, and that's the real challenge, I think, for the future. So the kind of questions that we're trying to answer now, we want to identify biomarkers to predict which cancers and patients will respond. There's a tremendous amount of effort in this space and some progress being made, I must say. What do we offer for patients who develop acquired resistance to anti-PD-1 therapies? We're starting to see that. Tumours are mutating and trying to interfere with that axis. How do we increase the proportions of patients who respond to anti-PD-1, PD-L1? There are lots of other checkpoints, as I showed you, and therapies in development. What other therapies do we use to treat cancers with microenvironments that are resistant to anti-PD-1, PD-L1? We have a novel immune checkpoint that we've discovered and patented, and we're developing that for clinical trial at the moment that we're hoping will be a good combination partner. What's the optimal scheduling? We're only really at the tip of the iceberg here in terms of optimising these agents. And how do we assess therapeutic index? I should say there are immune-related adverse events that occur with these particular therapies. They can be managed with steroids. But as we try and increase the curative potential of these agents, that's going to become more and more of an issue for these patients, particularly in the long term. And we're trying to model that, of course. And finally, I'd just like to thank the people involved in my career. This is the group of people that I'm working with currently. I want to pay specific attention really to my long-term collaborator Joe Trapani, who I built a cancer immunology program with at the Austin initially, and then the Peter Mac, fantastic collaborator that we really, I think, built this area up in Australia. And lastly, Robert Schreiber, who's been a long-term collaborator of mine, and the late Lloyd Old, who was really the godfather of tumour immunology and believed it and kept a lot of us going in those dark times. So thanks very much.
Thank you, Mark. Any questions? Time for a few questions.
Hi, fantastic talk. Can you expand more on how you think we can tackle that big problem that you described of immunological ignorance?
Yeah, there's lots of different ways to think about that. I guess there are people that are interested in T-cell therapies that want to try and get T-cells into the tumour more effectively, so that's one strategy. One of the things we've thought a lot about and we're working on now is trying to create an ectopic lymph node, if you like, in the tumour microenvironment. We think if we can do that safely, we can probably get immune engagement and get good effects there. So we understand a lot about lymph node organogenesis now from lots of developmental immunology. So the hope is that we can create that microenvironment in the tumour. And then there are other cell types which we really haven't worked a lot on to get effective tumour immunity. So a lot of tumours have neutrophils and macrophages, and they can have and granulocytes. They can have very powerful anti-tumour effects. So because immunology is very focused on adaptive immunity, I think there's a lot of capability of innate immunity to do the job as well.
If there are no other questions, please. Oh, yes, there is another question.
Are these treatments applicable to brain tumours? I mean, do they cross the blood-brain barrier?
Yes, they do. There's early data in glioma, glioblastoma, so and quite promising data for a subset of patients there. So, yeah, these therapies, the immune cells can effectively migrate and proliferate locally, and seemingly with the therapies, there hasn't been any, specific adverse events. Obviously, there's a lot of sensitivity about trying to treat people in the side of the brain because of the potential dangers of inflammation, but it's very early days there, but I think there's promise.
Okay, thank you very much, Mark.